Thanks for this morning. Report on our breeding project. It's been a two-phase project for the last classically breeding grapes, and sort of breeding technology has been applied to that. Your Mayor Aaron Howard wants to be our next your grape breeder, and she'll be taking over quite a bit of this project as time goes on. I have retired last June, and my replacement is not yet in place, but it's getting closer. So we're hoping to weave him into the whole process as well, him for as it goes. So with that, this work couldn't have been done without Samara, of course. And Alan Tencher, my assistant breeder in the field, Nina Romero was instrumental in all of the greenhouse testing and greenhouse management. It's working on characterizing the resistance genes we found in this process, and a number of graduate students and technicians as well. And of course, thanks to the PD-GWSS and Rossi and Dot Chair monies as well. So PD resistance breeding has been going on for quite some time. Really, Lenoir or Black Spanish, the primary and dominant wet red wine grape in the South now, is PD resistant. It was created long ago as an accidental hybrid between Vitis estabalis. That's still the mainstay in terms of fruit quality, and it's not particularly great. Some of you may have tasted it. It's a tolerable port. It's not such a good red wine, but not bad. And hundreds of years of breeding also has gone into this, with work that culminated with John Mortensen and the release of Blanc du Bois. And Blanc du Bois is a fairly nice, it's been growing also across the South now. It's fairly nice and quite resistant PD wine grape, but its last been apparent as Cardinal. It's a table grape, and it really doesn't have much of a wine grape character to it. It is fairly resistant. And we were lucky enough to discover a single dominant gene and material that Harold Olmo had left in the collections, a Vitis arizonica from near Monterrey, Mexico. And it has a single gene for resistance, and we can produce with other materials quite freely and generate lots of materials. We've collected over 1,000 accessions from the Southwest U.S. now and northern Mexico that we hope has other alternative sources of resistance to, that Samara will be speaking a little bit more about. Next. So our objectives were to develop large populations that up to the 97% vinifera level in diverse and the backgrounds as well. We wanted to integrate in different forms of PD resistance and stack them if we could, and that required mapping and understanding how these genes work and where they are in the genome. And we have a long ways to go with that, but we're moving very rapidly in that direction. Part of the beauty of looking at these materials closely and genetically is to look at their potential for genetic mapping. And we were able to develop markers, genetic markers, that we could use for screening these plant materials. And we use them as excluders, in essence. If plants don't have the resistance genes in them, we throw them away. But we don't really select for resistance until we've heavily tested these things in the greenhouse. And we're really sure that they're quite highly resistant. And there's quite a bit of variation, even though appears to be a single locus. It's quite complicated. And then we can take these materials that are 100% wild species and cross them with vinifera and end up with a generation that's about 2% vinifera. And as we go through this, the lineages, each year we get a little bit closer to 97%. One of the limitations in breeding these materials is the temptation to start making wines and evaluating wines at early stages in the selection process. And we knew that the wines wouldn't be any good until we got close to 100% vinifera in terms of standard quality. So we didn't waste a lot of time evaluating wine quality. We evaluated fruit quality, did organoleptic advancements for these things. But we really haven't concentrated on wine quality until the last two generations, which has been, I think, really important. And you can't really make progress without having this rapid 2-year cycle from seed to seed, and having the ability to accept that the fruit quality is not going to be great, so making wine would be a waste of time. We did make wines to prove the fact that was a waste of time, but it was a way of going through these things very rapidly. And so we have 5 releases out now at this point, Caminante Blanc and Embudo Blanc are the 2 white wine grapes. There are 97% vinifera. They have different viticultural and phenological and enological characteristics, and you can read that you're on board. We'll try to catch up some of our time there as well. But there's quite a bit of diversity, and it's fascinating as you're making these generations of back crosses. Once you get to about 88, 97% vinifera, the plants actually start looking more like vinifera instantaneously almost. And they are quite wild looking in the first generation. And you can clearly select for those too. The teeth are very different. The vine growth is different. The architecture of the clusters is quite different. So once you get up to these 94, 97% levels, they're indistinguishable from vinifera. You can't see any of those hybrid traits. And most of the unpleasant hybrid characters that are carried at the earlier generations in terms of wine quality also are gone at this point as well. Hard not to look at the babies here. They're beautiful plants. Here's Ambulo Blanc, different productivity, different other wine qualities. Caminante Blanc is really the most like a Sauvignon Blanc. And in tasting panels, it's always come out as indistinguishable from Sauvignon Blanc as well. And the Ambulo Blanc is a bit more like Chardonnay, more neutral in terms of aromatics, but very nice as well. But the first of the reds that we've released, this one is a somewhere between Pinot and Zinfandel, probably. It's quite a nice grape. Next one coming out is one of the few that we have at the 94% level, not the 97% level. It has excellent tannin structure and phenolic, and has a bit like Petit Syrah. And Ante Noir is the most tannic of the ones in this series of releases, and it's fairly productive. It will be a good blender, and it looks like it has nice aging potential as well. Next one. We have quite a few other plants that are cleared testing at FPS and certification FPS and are in process now. So there's a couple of them just to see some more whites and some more reds. Let's look at the next one. And another one of the red wine grapes. And you can read up on these too as we go through. And another of the red wines as well. And next one. These plants have been growing in PD affected areas. You can see here along the St. Helena area, the Napa River, Clarion Corridor, and all of the pot marks in there. And here you can see this band of perfectly healthy looking plants. Looks like it's a new road essentially in the picture, as you can see for the arrows. And that's all of our resistant materials. And they've been growing there for quite a few years at this point. It's really no sign of fierce disease, and they're holding up nicely. So as we go through, we've been field testing these materials and looking at them. They've been made into commercial wines at Camus at this point. And Whitehall Lane, some commercial winemaking has been done in Texas as well, because it's scattered in quite a few different areas. And it looks like they have pretty high potential. And so with that, to get us back on track here, I'm going to send you over to Dr. Riaz, who is fresh back from Pakistan and still probably slightly jet lagged, not too badly. And she's going to bring up to date on a bit more of the gene identification and genetic diversity aspects of the project.
Thank you so much, Andy, for the kind introduction. I will be continuing and starting my career at USDA ARS facility as part year, and where my focus is both for fierce disease resistance as well as horticulture. So I will talk with both about in coming slides. So as Andy mentioned, that we have collected over 1,000 sessions, and so they are mostly from Southwest. That also include the germplasm, which Dr. Omo collected from Mexico in early 60s. So most of this germplasm, it belongs to different Native American, Gardenia to Vitis amazonica, Mustanghensis, Rubestris, and Rasteria. So in upcoming slides, I will be talking about how we use this germplasm to understand and gain more better understanding of fierce disease resistance and its diversity. So our main objective is to identify the alternative resistance loci. So in this way, we can expand the genetic base of resistance. So for that purpose, so all the germplasm we have collected in last 20 to 25 years, it was screened for PD resistance using our greenhouse screen. And we are working with highly resistant germplasm only for our breeding purpose. When I say highly resistant, that means that it has a very low amount of bacterial titer. So all the germplasm, so we can do the genotyping with them. So in this way, we can carry out the genetic diversity parameters, because we do want to work with the germplasm, which is genetically different from each other as well. So the dendrogram of the right-hand side, that these are our sections which were highly resistant in multiple in greenhouse conditions. And so the different colors, they represent their different geographical locations. Like all the germplasm sections in red color, they were collected from Mexico. The one in blue, they were collected from Texas. And purple is Arizona. And there's one session, I was just going to call it Arizona. So, and one session, SC36, was collected from Southern California. So genetically, based on the nuclear SSR markers, they are all distinct from each other. They were collected from different locations. So in next phase, we developed small breeding populations, and we screened those breeding populations for PD resistance, also based on our greenhouse screen. And we already know that in the session B4317 at the bottom in the diagram, that's the one which we use to identify major locus CDR1. So now our main task is to identify additional loci, and for that purpose, we use the limited mapping strategy. So for that, so we have the phenotypic data from the small breeding populations, and we genotype them with the markers from chromosome 14, where the PDR1 locus reside, and we do the QTL analysis to determine if the marker has any association with the phenotypic data. So in next slide, I'm showing the main results. So all these sections you can see on the map, they are coming from collected from different locations. However, all the results, the QTL analysis with the small populations, it indicated that the majority of them, not the majority, with the exception of two sections, all of them, we identified that Pierce disease resistance is on chromosome 14 in the same within the same genetic window where we identified the CDR1. So the results of major results of this work has been already published in 2018. That paper was in TED. So D4226, that's a very unique accession which was collected from Baja California. So this accession is extremely homozygous, and it was quite difficult to find the markers for that. So there we identified another major locus on chromosome 8, which we named PDR2. And just recently, I completed the genetic mapping, limited mapping with ANU 67 accession. And so far, the results were inconclusive. That means that for this particular session, it is a potential candidate for further work, and potentially there are other loci other than the CDR1. So our main purpose is to broaden the genetic base of Pierce disease resistance, because right now our whole breeding program is focused on PDR1 locus from B4317, but now we have PDR2 from B4226, which is on chromosome 8. So we have already combined of PDR1 and PDR2 in three sections, which Andy mentioned earlier in his slide. So those are the three selections where both PDR1 and PDR2 combined, and they are already through FCS, and we are in the process of making small batches of wines to check the wine quality. ANU 67 is the other potential source of PD resistance, which is different from PDR1, and more work is definitely needed. And I think that as I start my career with the USDA, that will be again like one focus to continue work and help us to identify any other locus other than PDR1 and PDR2, which we can use in our breeding program. So apart from the breeding purpose, we wanted to also understand that how within this germplasm, which we screen for PD resistance, that what is the genetic level of genetic diversity of PD resistance, as well as we wanted to see that how the southwestern and southeastern germplasm differ in the PD resistance from each other. So for that, we did the genotyping with 20 SSR markers, and we genotyped 326 grape accessions that represent the grape species from Southwest as well as from Southeastern USA and the germplasm from Mexico and Costa Rica. So including the Southeastern germplasm was important because in earlier parts of PD resistance breeding, many breeding by more consent, they were using the Southeastern species and many hybrids were developed. So out of 326 accessions, 266 we tested them under greenhouse-based PD screen data, and we also included the 12 resistant hybrids based on the literature, the resistant hybrid cultivar that were developed from the southeastern grape species. So all the results of this work just published last year in POC 1. So the key finding of that, based on the genotype data, the both Bayesian clustering and the TICO-A analysis, it identified two main groups. So essentially, the southwestern grape species are very distinct from southeastern USA grape species, and it seems like the Sierra Madre Oriental that like present physical barrier for merging, and then definitely the Texas area is a hybrid zone where species hybridization is happening. So the Pierce disease resistance also followed the same pattern that the germplasm from southwestern USA had strong resistance to fierce disease compared to southeastern USA. And the hybrids, the 12 hybrids, known hybrids, which were the results of the breeding programs using southeastern USA grape species, they were not resistant at the same level as we have found in the southwestern germplasm. Also, we identified that the Vitis arizonica, that is a main species which showed us very strong resistance to fierce disease. So for both PD and switching gear to a little bit of powdery mildew work introduction also. So for both PD and powdery mildew breeding, we are following an integrative approach, which is that our main objective is to gain durable field resistance. So for this purpose, we are always in search of unique germplasm, identify the resistance, and develop markers which can be used for the breeding purpose. At the same time, it is also extremely important to determine the mechanisms of resistance so we can combine different mechanisms of resistance for this pathogen and then also understanding the pathogen biology. Here, our all colleagues were working with the bacteria and also with the insects. That information is crucial. So combining all this information together, that is the only way for us to gain durable field resistance for both PD as well as for target mildew. So I want to briefly give the outlook on our powdery mildew resistance breeding at UC Davis. We have made a tremendous success in that. So for here, we are working with multiple resistant loci. We have Muscadina rotatorifolia. From there, we have run 1, not 2.1. And then we have Asian Vitis vinipra, which is run 1. Other than carrot sandals, we have multiple other extensions which also carry run 1. And then we have Asian Vitis species, Vitis provenetiae, Vitis CSSPI, so which contributed REN 4, REN 6, and REN 7. So altogether, there are 6 major loci which we are working with at UC Davis for powdery breeding. And at this time, we have stacked lines up to 91 to 100% verifying them. Like we have different combinations, REN 1, REN 4, REN 1, REN 1, and also 3 loci from the same select scene session. So this is a tremendous resource which can be used down the road for combining PD and powdery mildew loci together. So now a little bit that what will be my objective at USDA Pioneer. So I have gained with working with Dr. Walker 20 years of experience of working with PD resistance and expertise and knowledge. And this, all the germplasm, it would allow me with the help of Andy that we can really put the table grape and raisin grape breeding at fast track and at Quartier Station. I know that the CDFA is more focused on the wine grape breeding, but they are important component as well because all the Central Valley they contribute to the grape industry as well. So at the same time, as we have already great resources for PD resistance, but for powdery mildew, also we have unique already stacked lines, and it is so much easier to combine PD and powdery mildew resistance together, even for the CDFA funded project as well. And in this way, for both diseases, they will be tackled within the same resistant line. So for this part, I will be definitely collaborating with other great leaders at ARS facilities as well as at UCG, and to put the molecular breeding at Parlier Station at fast track. For this purpose, we are already undergoing breeding strategy, and we are working. So it's just not just the disease resistance, but at the same time, if we want to put our release of new selections and varieties, we need to focus on other traits and other desirable traits for the wine quality, the fruit, other horticulture traits, and for both for wine, table, and raisin grapes. So by doing this, we will see the results which we have achieved right now with the 10 years, 10 to 12 years time, that rate can be made much more faster. And so on this, I will conclude my talk. Thank you so much. Good one. Wow.